Hello everyone and welcome to this session. My name is Ines Costa.
And my name is Jennifer Young. And this presentation is going to follow on from the one you've just heard. And we're going to take you through the next steps in translation.
And our talk is entitled From Radiochemistry to First Images, In Vitro and In Vivo Testing.
So before we start, we just need to state that neither of us have any financial interest to declare. So let's get started. If a radiotracer has been designed for use in humans, it needs to be tested to understand how it behaves in a biological system. We need to understand whether it needs to be adapted to give better targeting or imaging properties, or if these are fit for purpose. Often there's a number of development cycles that we go through before the best radiotracer candidate is selected. And biological testing, both in vitro and in vivo, are very important for selecting the best candidate.
So it makes sense to start with the simplest biological models and build up to those with more complexity, and our presentation will follow that trend. So let's start with cell culture. Cell culture is growing well-characterized cells, either murine or human, either as a single layer or in suspension, in a controlled environment. With this level of control, it is possible to design experiments that allow you to understand how radiotracers interact with cells and specific receptors or transporters on their surface.
This allows us to answer questions such as, are radiotracers taken up by cells and how is this mediated? Do radiotracers target the receptor or transporter that they are designed for? Do radiotracers behave differently in different cell lines? Do radiotracers get taken up in non-target cells? And what is the affinity of a radiotracer for its target receptor?
And for therapeutic radiopharmaceuticals, we can answer to the questions such as, can radiopharmaceuticals induce toxicity in form of DNA damage and cell death?
So here are some basic examples of how studies using cell culture can be used to assess radiotracers. Cells can be suspended in an epindorf, and then the radiotracer is added and incubated. Then cells are spun to separate the supernatant and the cell pellet, and the amount of radiotracer in each of these two fractions is measured. This allows you to compare uptake in different cell types, for example, those with the receptor and those without. And you can see an example of this in the graph. You can also use the same method to measure uptake over time for different radiotracers. Or if you're interested in the different affinity of radiotracers for a target, you can use assays such as an IC50 assay shown here, or a Ki assay, which helps you to determine the relative affinity of a radiotracer. Here you use a non-radioactive analog, which blocks binding sites and stops the uptake, allowing you to assess the IC50, the inhibitory concentration.
But when developing radiotracers for molecular radionuclide therapy, we also need to do in vitro tests to verify if the radiotracers induce toxicity. How do we do this? One of the assays that we use to test if the radiotracer induces radiotoxicity is the chlorogenic assay. This assay determines if the radiotracer can induce cell death by stopping cells proliferation. This means that while normal cells can grow and proliferate to form colonies, as you can see here, cells that are killed by the radiopharmaceutical cannot divide and form colonies. So here you can see that when cells are treated with radiopharmaceutical, they didn't grow to form colonies, as you can see, compared to the untreated cells. But we also want to test if the radiotracer can induce nuclear double-strand breaks, because these lesions are highly cytotoxic. This is because they are difficult to be repaired, and if they are prone to be misrepaired, leading to the cell death. As an example, you can see here in blue the nuclei, and in pink the double-strand breaks.
So once parameters of interest have been established in 2D cell culture, tests can be done in more complex models. Examples of this are spheroids and organoids, and here you can see the distribution of radiotracer uptake in either of these models.
We can also test the radio tracer in isolated organs, such as an isolated perfused heart. And this can also provide us a well-controlled situation to test the new radio tracers.
This leads on to in vivo testing, experiments involving animals. And during radio tracer development, this focuses on testing in mice and in rats. It's important to note that the UK has some of the highest standards of lab animal welfare in the world. All research in the UK must be approved by the Home Office, and researchers and the institutions doing research must be licensed by them. Also, ethics committees exist to ensure that the potential benefits of research outweigh any suffering to animals, and animal research can only be carried out when there is no suitable non-animal alternative. If you'd like to find more information, please look at either of these websites. An important principle that underlies animal research is also the 3 Rs, which stand for replace, reduce, and refine. And so all animal experiments that take place should think about these principles when designing experiments. Imaging is a non-invasive technique, and so it allows us to actually reduce and refine the number of animals that are used. So by developing new imaging techniques for preclinical models, we also reduce the number of animals that are used in other forms of research.
Testing of radio tracers in animals normally involves the administration of the radio tracer through the same route you like to hope to deliver clinically, for instance, intravenously, and then monitoring its biodistribution over time in both healthy animals and in diseased animals. This allows us to answer questions such as:
what is the biodistribution and its excretory pathway for the radio tracer? How does this change over time? Does the radio tracer accumulate at the disease sites? What information can you obtain about the disease by using this radio tracer? What is the optimal time for imaging to get the best target to background ratio? And for therapeutic radiopharmaceuticals, can radiopharmaceuticals reduce disease burden? And can they slow progression of disease?
And here you can see examples of small animal scanners suitable for imaging of mice and rats.
In the example of imaging healthy animals to understand biodistribution, the process is that the radio tracer is administered to the animals are placed onto the bed, and anaesthetic is delivered through the mask. Then the CT and the PET or the SPECT can start. The animal's temperature and breathing is monitored throughout the process. After scanning is complete, the images are reconstructed according to the scientist's protocol and are ready to be viewed. In the example image here, you can see the radio tracer has accumulated in the gut and in the bladder. You can then go on to analyse the images using software such as Vivo, shown here. Images captured at different time points can be analysed. You can draw regions of interest and you can extract different types of data.
If required, the Symmetry data can be determined from imaging data, which help us to predict the effective dose in humans and observe those to key organs to determine the expected safety profile when translated to clinical practice.
Disease models can also be used, the most common of which is a subcutaneous xenograft. Here, the cell line used for in vitro testing is injected under the skin of immune-compromised mice, and a tumour grows at the site of injection. The imaging agent can then be injected, and the uptake of the radio tracer into the tumour can be determined.
As well as cancer models, there are lots of animal models that can be used to assess the radio tracer behavior in a wide range of diseases.
Again, images can be analysed to compare different organs or, in fact, different tumours, and the performance of different radio tracers can be assessed.
So that brings us to the end of our talk. We hope this has helped you understand more about the in vitro and in vivo testing of radio tracers and the steps required to move towards translation to clinical practice.
So thank you to all of our colleagues that provided figures for this talk, and thank you all for listening.